Hi, I'm Tom. This is Davey and David, and we're working on few shot classification of target activity augmented with pre-trained protein embeddings. To give some context to the work we're doing, we're working in the drug discovery space, and drug discovery is notoriously difficult with a high error rate and a high failure rate. One of the key steps of this process is hit identification and lead optimization, which is essentially finding a small molecule, which will end up being a drug that binds to a biological target or protein of interest. Due to the lack of high quality experimental data that plagues the drug development space, few shot approaches for these tasks are really relevant and exciting. And in fact, some previous work has explored few shot approaches to binding prediction between a ligand and a protein. However, none of these works have incorporated any data about the protein target being bound, only the ligand. So the novelty of our work is including protein information, which we generate through a pre-trained protein language model that takes in a protein sequence and generates an embedding into our downstream models to improve the accuracy of our binding prediction tasks.
So our meta-learning problem is 5-shot two-way classification of ligand protein binding. Each task is composed of one target protein, 5 support examples of binding ligands, 5 support examples of non-binding ligands, and a query set of 1 binding and 1 non-binding ligand. With regards to embeddings, as Tom had mentioned, we use previously published transformer-based encoders, namely ChemBerta and ESM2, to generate embeddings for all our proteins and ligands. We experimented with an LSTM-based black box meta-learner, a BERT-based black box meta-learner, and a prototypical network, as well as 3 different ways of feeding protein ligand SMILE embeddings into our meta-learners. The first of which is to only use the ligand SMILE embeddings and withholding any protein information as a baseline. The second is concatenating the protein embeddings to our SMILE embeddings from the very beginning. And then the third, which is specific to our black box meta-learners, is to introduce the protein embeddings at an intermediate step of black box meta-learning, namely between the 2 LSTM layers or between the BERT encoder and the linear layer.
Yeah, so we evaluated our model using 5-shot two-way classification accuracy on a held-out meta-test set, and these results are in this table here. Our baseline performance using just the SMILES embeddings for the molecules hovered around 65 to 66% for our LSTM and BERT-based black box models in our protonet. And in the 2 rows below here, we're showing accuracy performance incorporating the full 640 dimensionality ESM2 protein embeddings. And here we're concatenating those embeddings before the model. So in this case, that would be down here. And here we're concatenating either after the first LSTM block or after the BERT encoder. And in general, we saw that concatenating before led to better performance than concatenating after, potentially because the black box model is able to get more expressive with these, how it's using these protein embeddings, as opposed to concatenating them afterwards. And we also found that we were able to significantly reduce the dimensionality of the ESM2 protein embeddings using a VAE down to a dimensionality of 50, and still retain a pretty significant portion of the predictive performance, as shown in these 2 rows below. And specifically for our VAE, we found that we were able to reduce down to a dimensionality of 50 and retain a reasonable MSE loss, which was particularly better for the protein decoder than the SMILES decoder. So basically, all of these results go to show that using these protein embeddings, and especially with a black box model, we're able to sort of better generalize to new protein prediction or affinity tasks in the meta-test set. And this could indicate, for instance, that the model is able to pick up on certain patterns between different protein targets. For example, similar types of proteins or similar electronegativities or even other protein characteristics. And it's able to sort of incorporate that information into its few-shot classification of drug interactions, which is a finding that, to our knowledge, hasn't been demonstrated in prior literature. So yeah, that is our project, and thank you for listening.